The cells that process and present antigen are going to present that antigen to T lymphocytes or T cells. Those T cells will actually interact with the antigen. They will actually bind with the antigen being presented. And remember, anytime a cell binds to another molecule, anytime there's an interaction between the receptor and the thing that binds to it, some process is going to be activated. These T lymphocytes are only going to be able to bind to antigen that has been correctly processed and correctly presented. And that's a good thing, because that will help protect us against abnormal immune reactions. Once a T lymphocyte is presented with antigen and actually binds to that antigen itself, that T cell is now said to be sensitized to that antigen. It has recognized it, and that process is going to change that T cell. It's going to cause it to mature and become a slightly different type of T cell. And there are many different types of T cells, and you may have heard of some of these. There are what are called T helper cells, and there are what are called T cytotoxic cells, and so on and so on. It's not important that we know the names of different types of T cells. It's more important that you understand that it is the T lymphocyte that is the type of cell that the antigen is presented to. And it's the interaction of that T lymphocyte with the antigen that's going to set the adaptive immune response into play. Now, of all the antigens in all of the world that are going to get presented to these T lymphocytes, we can break them down into two classes. We can break them down into what are called exogenous antigens and endogenous antigens. Now, why is this important? Well, a couple of reasons. Number one, exogenous antigens are specifically what bacterial antigens belong to. Exogenous means coming from outside, exo. If you think about it, the antigens that are on a bacterial cell are literally coming from the outside world into the body, if that bacterium is a pathogen. So the antigens that that bacterial pathogen is carrying, it's being brought into the body from the outside world. Endogenous antigens are different. Endo refers to within. So these are antigens that are coming from within the body. And these are the types of antigens that viral proteins are. Remember, viruses are obligate intracellular parasites. So when viral particles come into the human body, they have to invade our cells in order for infection to occur. So once they get into our cells are going to copy their genetic material and express the viral genes. And that leads to the production of lots of new viral particles inside that host cell. Now the host cell is inside the body. So it's not until the new viral particles get released from the host cell that they're recognized. Let me say that again because that's important. The host cell is already in the body, and the viral particles will be released from the host cell. And that process is what triggers the immune response. So those antigens, the viral proteins, are coming from within the body. They're coming from our own cells, our host cells. And they are considered endogenous as a result. Now, why does all this matter? Why is it important that bacterial antigens fall into the exogenous group and viral antigens fall into the endogenous group? Well, it turns out that the antigen processing receptors, the MHC molecules, are going to vary for the bacterial antigens and the viral antigens. There are MHC molecules, in other words, antigen processing receptors, that are called class 1 because they're going to present endogenous antigen, viral antigen. And then there are MHC class 2, which are going to present exogenous antigen or bacterial antigen. Two different classes of receptors on our macrophages and our dendritic cells that will present antigen to T cells. Take a look at this slide now. On the left-hand side, we have a drawing of this really important process. If you follow through the numbered explanations for the diagram, you can watch this antigen processing occurring. Now, this drawing represents a macrophage, and it's a pretty good representation of a macrophage. They are not the kinds of cells that we typically imagine in our mind, having a nice smooth circular or spherical shape. Macrophages have a very irregular shape. Their membrane is not a nice, pretty, smooth surface. It's got bumps and crazy projections coming out of it. Now, what's happening is we have a bacterial pathogen that has breached a barrier and has initiated an innate immune response. And those bacterial cells are represented up here by these green rods. Now remember, the sentinel cells are going to interact with this bacterium and are going to trigger inflammation. But a couple of those sentinel cells are also going to process the antigens that are in this bacteria. So that's what this macrophage is doing. It's going to actually engulf, it's going to actually consume a few of these bacterial cells. That's how macrophages got their name. Word macrophage translates as big eater. I love that. Macrophages are physically going to take these bacterial cells inside and then break them apart and process the antigen. Now, what you're looking at here in the pink color, this is a type of organelle called a lysosome. Remember, eukaryotic cells contain organelles, and the lysosome is an organelle that helps break down and process these bacterial cells. Lysosomes secrete enzymes, and the enzymes will break apart that bacterial cell into its component molecules. Now, what you're looking at over here, drawn in blue, are MHCs. So these are the molecules that are going to act as receptors on the surface of this macrophage. So what's going to happen is the bits and pieces of that bacterial cell that are going to be the antigens, they'll get attached to the MHCs. Now, in this case, these are MHC class 2 because they're bacterial, they're exogenous antigens. Now, it's really hard to see, I know, but the antigen is depicted as a very small yellow circle here. And that yellow circle is going to get attached to this blue receptor. And the combination then will be inserted into the membrane. So now that antigen, that processed antigen, is sticking off the surface of this cell so that it can be presented to the lymphocytes, the T lymphocytes. Now take a look over here on the right-hand side at this amazing image. This is an image that was taken with a scanning electron microscope. What you're looking at here is antigen presentation. This is the dendritic cell. The dendritic cell that is one of the two types of antigen processing cells. Now you can't see the MHC here. You can't see the little piece of the bacterial cell that's acting as the antigen. But what you can see is that this dendritic cell is attaching to this lymphocyte, this T lymphocyte. And the attachment point is the antigen that's being presented. There's an MHC on the surface of this dendritic cell, a receptor that has a piece of that bacterium attached to it, the antigen. And the lymphocyte, the T lymphocyte, also has a receptor on its surface that is going to bind to the antigen that's being presented. It's like the dendritic cell is reaching out with an arm and a hand, and in its hand is an antigen. And the T lymphocyte is reaching out with its arm and its hand, and it's going to take the antigen that is being offered by the dendritic cell. That's what this drawing shows us. So, thinking back to that electron microscope image, this is the antigen presenting cell. It's either a macrophage or a dendritic cell. This is the T lymphocyte. Off of the surface of the antigen presenting cell, there's an arm with a hand. That's the MHC. And in the hand, there's an antigen. A protein, perhaps, that was harvested from that bacterial cell. The T cell, the T lymphocyte, is also reaching out an arm. That's its T cell receptor. And it's going to bind to that foreign protein. So these two cells are both reaching out a hand, and the hands are holding on to this foreign piece of material, this foreign protein that came from the bacterial cell. This is antigen presentation. The antigen presenting cell is presenting the antigen to the T lymphocyte. So what is triggered by antigen presentation? Well, the response to antigen presentation is twofold. Number one is a cell-mediated response, and number two is a humoral response. The cell-mediated response is that the T lymphocytes, as I said a minute ago, are going to mature into other types of T lymphocytes. So that receiving T lymphocyte that we just looked at in the image is going to go on to produce more mature forms of T cells, T helper cells and T cytotoxic cells, and so on. The T lymphocytes are also going to secrete some humoral chemicals. The humoral part of the response, the non-cellular part of the response, is going to be the production of antibodies. Now, this involves a new type of lymphocyte that we haven't mentioned yet. That's called a B lymphocyte, or more simply, a B cell. So there are two critical types of lymphocytes that are going to get involved in the adaptive immune response. There are T lymphocytes, or T cells, and there are B lymphocytes, or B cells. The T cells are going to respond to antigen presentation with both a cellular response and a humoral response. The cellular response is differentiation or maturation into different types of T cells. And those T cells themselves will go on and release humoral chemicals. The humoral chemicals that get released by these mature T cells have a job to do. And unlike the humoral chemicals we've talked about so far that elicit inflammation, these humoral chemicals have the job of activating B cells. Now, what's going to happen to the activated B cell? Well, it's going to mature. It's going to differentiate. The B cells that are activated by these humoral chemicals are going to start dividing. They're going to produce two new types of cells, what are called plasma cells and what are called memory cells. Plasma cells will turn around and start making antibodies. Memory cells are going to do just what the name suggests. They are going to retain a memory of the antigen that started this process. And what will happen is those memory cells will be able to recognize that antigen if it ever comes back into the body. Those memory cells will be able to recognize that antigen immediately. So the next time the antigen is encountered in the human body, the memory cells will immediately respond and stimulate antibody production. So the second time the pathogen invades, and the third time, and the fourth time, and however many times the pathogen tries to invade, antibodies will get produced much more quickly. And that pathogen will end up being destroyed much more quickly. Take a look at this diagram that sort of summarizes this adaptive immune response. We're going to start right in the middle of the diagram right here, and I'll talk about this in just a minute. Here is the T cell. The T cell that has interacted with the antigen presenting cell, either the macrophage or the dendritic cell. This T cell was then stimulated to mature, and it matured into a particular type called a helper T cell. Remember, there are other kinds of T cells as well. We don't need to focus on the variety of T cells. The important thing is that this newly mature helper T cell is going to then turn around and activate a B cell, which is represented here on the left. T cells do that activation by releasing humoral chemicals and also by physically interacting with the B cell itself. You'll notice that the B cell also has MHC on it. This is where it gets a little confusing, as though it weren't confusing already, but it's important to know that all cells inside the human body have MHC. All cells have MHC. MHC1, MHC2. So yes, even B cells have MHC, and that MHC can get brought to the surface of the cell and can interact with antigen. Now this is the point where we'll go up to the top of the diagram. And this is the point where I'm going to remind all of us that none of these immune processes are happening all by themselves. All of this occurs in an intertwined way. So while this adaptive immune process is going on, There's also just pathogenic bacteria around, right? There are sentinel cells that are still interacting with bacterial cells, and there's inflammation going on, and so on and so on. So it means that in other places there are bacterial cells that are also interacting with B cells. And I don't want you to get too bogged down with this detail, but just understand that B cells, just like we saw a minute ago with T cells, B cells are able to interact with bacterial antigens and bring their MHCs to the surface. That's how this happens. Here's the MHC over here in white on the T cell. Here's the MHC over here on the B cell, and both of those receptors are recognizing this antigen. Meanwhile, this T cell is releasing chemicals, humoral chemicals, that are enhancing all of this, activating this B cell and causing it to start dividing. That's the really important take-home here. The differentiated, mature T cell is going to activate this B cell. And that activation is going to cause it to divide. It's going to produce two very different types of cells as it divides. It's going to produce what are called plasma cells. Those are these over here. And it's going to produce what are called memory cells. The plasma cells are going to start secreting antibodies. And all these little gray Y-shaped things over here, those are antibodies. These antibodies are very specific. They are targeted specifically against this antigen that helped make them. Plasma cells are going to make antibodies, and antibodies are going to help in the fight against this pathogen. Memory cells have a different job to do. Memory cells have to be able to remember this antigen. And the way they do that is by binding to and sort of exposing these antibodies on their surface. These memory cells now, the next time these cells come in contact with this antigen is going to bind to these receptors, these antibody receptors. And that will awaken the memory in this cell so that it can then stimulate more antibodies to be produced by plasma cells. In other words, the second time this antigen comes into the body, it's going to meet these. And the second time the antigen comes into the body, it will bind to these receptors on these memory cells. And it will end up in the production of antibodies much, much quicker the second time than the first time. So, the immune system is a very complex system, and we have only scratched the surface today. But remember, as you're going through this lecture and putting a set of notes together, focus on the key points of each of the three systems: the physical barriers, the innate immune system, and the adaptive immune system. In the last video for this lecture, we're going to finish up this topic by talking about antibodies and the key things that we need to know about those critical structures.